DNA and Biotechnology, Chapter 21. In this chapter, we're going to cover the form of DNA, replication of DNA, gene expression, mutation, epigenetics, and genetic engineering if we have time. We're going to start off by looking at the form of DNA. DNA is going to store all of our genetic information. It is the instructions that make us who we are. On its simplest level, DNA looks like long, thin strands. If you were to unwind it and stretch it out, every chromosome of DNA in your cell would exceed 5 feet in length. All of that's going to be packed into a tiny little nucleus. DNA is short for deoxyribonucleic acid. It is a double-stranded molecule that resembles a ladder that is gently twisted to form a spiral, or what is known as a double helix. Each side and half of each rung is going to be made up of a string of repeating nucleotides. A nucleotide is going to contain, they're relatively simple in structure, they contain a sugar and in this case the sugar is going to be deoxyribose, one phosphate group, and one nitrogen-containing base. So here are 4 bases that might be found on our DNA molecule. There are only 4 different types of nitrogenous bases that are found in DNA, from which we get all of the instructions for the rich and diverse life. Those 4 possible bases are going to be adenine, guanine, thymine, cytosine. Those are our 4 bases. The rungs of the ladder consist of paired, and each rung of the DNA ladder is composed of 2 complementary nitrogenous bases. The nitrogenous bases always pair up according to a few simple rules, known as the complementary base pairing. And that is that adenine only pairs with thymine, thymine only pairs with adenine, cytosine only pairs with guanine, and each pair of bases is held together by a hydrogen bond. Genetic information is going to be encoded in the sequence of bases.
The building blocks of DNA are units called nucleotides. Nucleotides are composed of 3 types of component molecules: phosphate groups, the sugar deoxyribose, and a nitrogenous base. There are 4 common bases: adenine, A, guanine, G, thymine, T, and cytosine, C. The DNA molecule consists of 2 separate chains of nucleotides, usually called strands. Within each strand, the phosphate group of one nucleotide bonds to the sugar of the next nucleotide in the strand. This bonding pattern produces a backbone of alternating covalently bonded sugars and phosphates. Each base extends from one backbone to the other to link up via hydrogen bonds with a complementary base from the other side. Base pairing is complementary, that is, A always pairs with T and G always pairs with C. The 2 nucleotide strands in a DNA molecule are wound around one another to form a double helix. When a cell divides, all of the DNA must first be copied so that each daughter cell will have a complete set of genetic information. During replication, the 2 strands of DNA that form the double helix separate, and each now serves as a template strand for replication. Free nucleotides base pair with their complementary nucleotides on the existing template strands. The result is 2 identical strands of DNA. Each replication process combines an old strand with a strand made of new nucleotides. That is, each one has a parental strand of DNA and one newly synthesized complementary strand. This model of replication is called semi-conservative replication.